Good evening, all. Hello. 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 Saraswati. Hello. 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 Today topic is gene libraries. What is library in general definition? There are different type of libraries are there. Library means different type of tool storage. So book library, the storage, the books. So that's why here the gene library. So here the genes storage. A library is a convenient storage mechanism of genetic information. Let's see how to store it, where it is stored, which is two types. Gene library, two type. One is genomic libraries. Second one is cDNA libraries. Genomic libraries. A genomic library is an organism-specific collection of DNA covering the entire genome of an organism. For example, if we take human being, 46 chromosomes, the total genome content called as genomic libraries. cDNA library, it is the DNA copy of an mRNA molecule generated by reverse transcriptase, a DNA polymerase that can use either RNA or DNA as the template. Let's discuss one by one. First, genomic library. As I said, first genomic library. In genomic library, first thing, we take entire content of an organism genome. For example, this is the entire content of genome. Total content of genome. Take the total content of genomes and cut into number of pieces. Cut into number of pieces. So which enzyme involved in which enzyme involves in genetic gene cutting? Here the restriction endonuclease enzyme involved in gene cutting. So first, for cutting, restriction endonuclease, restriction endonuclease, restriction endonuclease use. With help of restriction endonuclease, this entire length of the DNA cut into number of pieces like this. This DNA fragment, this is the DNA fragment, this is the DNA fragment, right? After this breaks into number of number of fragments. Now this DNA insert into the proper vector. The common proper vector utilized here the plasmid, already completed the plasmid vector. If the content is smaller, we can use the plasmid vector. But the same way, if the content of the genome more than 3000 nucleotide, for example, this is the fragment. This fragment just to 100 to 150 nucleotide space. In 100 to 150 nucleotide, we can use the plasmid as a vector. But if the fragments exceed 300 and above, we can use another vector, such as we can use bacteriophage vector or cosmid as a vector. Cosmid as a vector or bacteriophage as a vector. But if exceeded 3000 above, 1000 to 3000 above, in that situation, we can use another vector, that is YAC. YACS vector or BAC vector. YACS vector or BACS vector. Right? YACS vector means yeast artificial chromosome. The same way bacterial artificial chromosome. Yeast or bacterial artificial chromosome. Right? So these are the vector. So, but of course here given plasmid is the vector, but the plasmid utilize the vector when the smaller fragment, just 100 to 150 nucleotide. If it exceed the number of nucleotides or the length of the fragment exceed, we can use another vector such as cosmid or bacteriophages vector. If it is more extended, in that situation, we can use the AC or BAC vectors. This first vector is YAC, yeast artificial chromosome vector, which is extracted from the yeast cell. And second one extracted from the bacteria cell. So anyway, we cut into number of fragments, and these fragments, for example, we are 1, 2, 3, 4, 5, 6, 7, 8 fragments are there. For 8 fragments, we selected 8 vectors and inserted into each one. Now this is called rDNA, recombinant DNA. Recombinant DNA. Just wait. Hello? Hello?
Hello?
Why did not join earlier? Any problem? Where is so late?
Sir? Good evening, sir.
Right. Today topic gene library. The library means collection and storage. Like that here also the different type of gene collected and storage. This mechanism of genetic information called as gene library, which is two types, genomic library, which is two types, genomic library and cDNA library. Genomic means what is genome first? Already completed. What is genome definition? Hello? What is genome? What is gene?
Functional unit, sir.
Functional unit of what?
Functional unit of DNA.
Functional unit of DNA. Functional segment of DNA, gene. What is genome? What is genome? No problem, say wrong answer. Gene, right? Active site, active segment of DNA, okay. Genome, entire set of genes of an organism. That is called genome. Genomic library means the entire set of genomes collected and stored. This mechanism called as gene library. A genomic library is an organism-specific collection of DNA and storage of entire genome of an organism. That is called genomic library. And another type of gene library is cDNA library. cDNA, already told you the cDNA which is developed from the mRNA copy, RNA copy. Actually, how the mRNA prepared, how the mRNA synthesized. Hello? How the mRNA synthesized. Am I audible to you?
Transcription, sir. Transcription.
Transcription. In transcription, what is the template stand to prepare mRNA? Hello? Okay, transcription. But in transcription process, how mRNA synthesized?
From DNA, sir.
From DNA. But what is cDNA here? Reverse. That means from the RNA stand to DNA synthesized. The DNA called as cDNA and the collection of that type of DNA, storage and collection, that mechanism called as cDNA libraries. Now let's see one by one. First genomic library, next cDNA library. In genomic library, right. In genomic library, we have to see two steps. One is construction of genomic library, screening of genomic library. Let's see how the constructed of this genomic libraries. First, as I said, the definition, the entire set of genome. Here, you selected the some total portion of the chromosome here. or gene, and which is cut into restriction endonuclease enzyme already complete different type of restriction endonucleases. Function of endonucleases. Another name of endonuclease, already told you, the endonuclease is called as molecular Caesar function cutting. So this gene cut into number of fragments, 1, 2, 3. Here we can see the different type of fragments. After cut into number of fragments, these fragments inserted into vector. Here the different type of vectors are there. The most useful vector is plasmid vector. Apart from the plasmid vector, bacteriophage vector, cosmid vector, VAC vector, BACS vector, they are all present. But if the fragment is small in portion, with made up of 100, 150 nucleotide, we can use plasmid as a vector. If the fragment, cutting fragment more than 300, we can use cosmid as a vector. We can use cosmid as a vector or bacteriophage as a vector. If it is exceeded more than 1000, in the situation we can use YAC vector, yeast artificial chromosomes vector, or bacteria artificial chromosome vector. These any type of vector we can use in genomic library. That is the based on length of the fragment, length of the cutting fragment. If the length of the cutting fragment small, we can use plasmid. Very larger, we can use YAC vector. So anyway, after plasmid with gene cutting fragment, this both combination called as recombination DNA or recombinant DNA or rDNA. Now this different type of rDNA stored in another vector, another host cell. The host cell should be, what is the host cell here? Hello?
Bacteria, sir.
Bacteria is the host cell because the plasmid present in bacteria. That's why this rDNA stored inside bacteria. So let me change color here. This is red color plasmid and the blue color fragment, cutting fragment. Blue color cutting fragment. Right? Now this bacteria take into culturing, into petri dish. In agar, nutrition agar medium, they take one bacteria first. It is replicate or due to binary fission, it forms a number of different type of bacteria. Like that, we can take, this is one plasmid went into bacteria. This is one plasmid and another bacteria. This is another bacteria. This is another bacteria. This is inserted another bacteria. This is also transferred into another bacteria. Like this, number of bacteria are formed with rDNA. This transformed bacteria. Now this bacteria called as transformed. Why it is called as transformed? Because the rDNA transferred into the bacteria. That's why it is called transferred bacteria. This transferred bacteria cultured in nutritional medium, agar medium. Due to some weeks, it is multiple in number. It becomes multiple in number. Now this is called construction. Construction what? Genes and stored. Where the genes are stored. Hello? What is definition? Genomic libraries? Libraries, collection, storage.
No.
Collection, storage, right? The collection from the fragments from the genome. First genome cut into pieces. Next, collected into the plasmid. After formation, rDNA stored inside the bacteria cell, right? Later, this bacteria cell, when enter into the, when give to the culture media, automatically it improve in number.
It undergo binary fission, sir.
Yes, binary fission. that in binary fission that can multiple in number. So this is storage. And now like that, the constructed the genomes, different type of genomes. Now scan the genome. Hello?
Sir.
Right. Now scan the genome. Now it is clear for construction. Construction clear, all of you?
Yes, sir.
Yes.
Right. After construction,
Screen construction of genomic libraries.
after construction, next what? Screening. 2 steps in genomic library and also cDNA library also 2 steps. One is construction, another one is screening of genomic library. And before construction, and there are some extra mechanism also from construction of genomic library. It's given in our book. Just cDNA construction. Okay, no need. No problem. Right. Now screening. What you screen here, first of all? In first step, you selected the entire set of genome because, for example, a plant has a high resistant power. Plant has high resistant power. You selected this high resistant power genome. This high resistant power genome. Cut into number of pieces. But here if you don't forget, you collected the entire set of genome, right? You cut into number of pieces of entire set, right? Inserted into vector, right? Now transfer it to the bacterial cell and culturing it in petri dish agar media. But you don't know where the desired gene. Can you identify desired gene after construction? After construction genomic libraries, can you identify in this group of bacteria cell, in the group of bacterial cell where the desired gene is present, can you identify? Hello?
No, sir.
We cannot identify in this much number of cells. That's why we should screen them.
By screening we can identify it.
Yes, by screening we can easily identify. Now, how to screen? How to screen? First, in screening mechanism, already some colonies are grown in nutritional media of petri dish, right? In this group of cells, first of all, we take here, this is colonies. Before colonies, we add some nitrocellulose filter paper. This membrane, this is, for example, nitrocellulose filter. Nitrocellulose filter, right? nitrocellulose. This technique called as colony hybridization technique. There are number of different type of technique are there to screen the desired gene identification. But we selected the best process is the colony hybridization, because we selected the vector is the plasmid. If you selected the vector as a plasmid, a colony hybridization technique is the best way to recognize the desired gene among the culture, right? The identification of cloned genes from vector or bacteria colony is an important step in the construction of genomic library, right? The desired gene DNA fragment from the genomic library can be recognized by the special method that is called colony hybridization. Colony hybridization. Colony hybridization method. Colony hybridization. It is a common method screening of plasmid based. Already told you earlier, we selected the vector as the plasmid. That's why this is the plasmid based genomic libraries. The host bacteria containing a plasmid based, right? Because already the plasmid we extracted from the bacteria. So that is the vector. Vector host is bacteria, right? In first step, to bacteria culture, nitrocellulosic paper covered on the culturing. Actually, this paper treated with alkali chemicals. Alkali chemical. The function of this alkali chemical disassociated bound of DNA duplex. How is DNA always double helix bond, double helix form, right? This double helix form with help of nitrocellulose, alkali chemicals, it is denatured into single strand. Now it is in the genome's DNA libraries, DNA present in double strand, like this. In each DNA, each bacteria, the genome with double stranded. When the covered with nitrocellulose paper, automatically with help of the action of alkali, it becomes haploid. That means single strand. Single strand. Single strand. Just compare here double stranded. Here double stranded DNA. After the action of nitrocellulose paper, it becomes single cell, right? Now next step, add labeled probe. Labeled probe to the single stranded DNA. The probe means here the complementary strand of desired gene. Already in probe, complementary strand present. A probe is used which can be a mRNA or homologous gene from another organism, synthetic. It is made up of oligonucleotide having similar sequence as desired DNA fragment. In probe, similar sequence of desired gene. But here in this group of gene, we don't know where the desired gene present. But in probe, the desired DNA sequences already added to the probe with oligonucleotide. Already told you oligonucleotides, short form of nucleotides, short length of nucleotides. With this probe, where after added into the culturing bacteria, what happened? What happened with help of probe? Already told you the probe already have the complementary sequences.
It helps to identify desired genes,
Right.
desired
Always the
bacteria.
complementary strand bind to the template strand. For example, in a probe, the complementary DNA sequence, guanine, cytosine, adenine, somewhat guanine, like that. Right? This is the actual DNA, desired DNA sequence. But this type of sequence, we don't know where this type of complementary sequence. Complementary means here the C, G, T, C. You don't know where this type of sequence present in this group, in this colony. That's why you selected earlier this desired gene sequence, complementary sequence with the help of probes. Now you add it to the culture. Now automatically this probe attached to the with help of this probes attached to the where desired gene, attached to the desired gene, right? Now we selected the desired gene, and where the desired gene present, that we have to select. Later, now this type of desired gene, you have to get number of copies through the rDNA technology, genetic engineering technology. Right? This is called as screening. Screening clear? Hello?
Yes, sir.
Yes, sir.
Any doubt in this?
Sir, visualization means once again, please repeat that one, sir.
Where the visualization?
Here, sir.
Yes, sir.
After
Incubate.
probes, labeled probes.
Yeah, the incubation with probes. During incubation, we get the number of desired gene. That means only we get number of desired gene. Now,
Sir,
once you check.
Sir, is this bacteria Bacillus or cocci, sir?
Escherichia coli.
Okay. Okay, sir.
Plasmid mostly we separate from the E. coli bacteria. But
Okay,
here
sir.
the visualization means whether the character expressed or not. That's what we have to visualize. Right now we get finally hybrid DNA. Finally we get hybrid DNA, desired character DNA. Right?
Yes, sir.
The next step. What is the next one? First method complete. Genomic library is completed. What is second step? cDNA library. What is cDNA? Already told you. What is cDNA?
mRNA to DNA.
cDNA.
Complementary DNA.
cDNA libraries. Right. In cDNA libraries, first, in genomic library, we selected directly gene, right? Hello?
Sir, here we have to. Sir, here
First
we have to.
mRNA.
Yes, sir.
mRNA.
Messenger RNA.
Yes. But we don't take the DNA because already told you DNA as active and inactive site, introns, exons. Do you know introns and exons? Hello?
No, sir.
Did you attend transcript? Did you attend a gene regulation class?
No, sir. I didn't attend that one, sir.
Eukaryotic transcription, eukaryotic gene transcription, eukaryotic DNA transcription. And DNA transcription, here the mainly template strand DNA synthesize mRNA. Okay, but this mRNA synthesized by active mechanism, which means actually in DNA both are present. Introns and exons are both are present, but introns non-functional. Only
Okay.
exons are functional.
Okay,
That's
sir.
why here the intron should be removed out. That is called spliceosome
Splicing.
mechanism, right? So anyway, the unwanted introns, non-functional introns removed out, and finally prepared a matured RNA, right? In RNA, only functional group present, only functional group. Almost if you take the entire genome, 95%, 95 to 98% junk DNA, not functional. But rest of the 2 to 5% only the functional DNA. In mRNA codes, only that type of functional gene. Now this mRNA in reverse process, what we get? DNA. What we get? DNA. What we get? DNA strand. Right? Here the enzyme utilized a transcription, here reverse transcriptase. Here the enzyme reverse transcriptase. Reverse transcriptase enzyme utilized. So as a result, DNA strand formed. Right? After formation DNA strand, how many strands this is? Double strand or single strand DNA?
Single strand, sir.
Yes, single stranded DNA formed. Later, with help of the polymerase enzymes and primes, this DNA becomes double stranded. That means this synthesize new DNA. Complemented DNA strands. Now, these DNA, same process inserted into where? Where it is inserted?
Sir, in cultural media, sir. No.
Before culture media. First,
Yes.
what is the vector here?
A host.
Bacteria, sir.
Bacteria, sir.
Bacteria is the host. That host required after formation. Okay, this is the, for example, this is the gene, some of the desired gene, group of desired gene segment, right? This desired gene segment actually with help of restriction endonuclease cut into pieces, right?
Yes, sir.
Break into fragments. Like that here, number of fragments are formed. Right? After this formation of number of fragments, these fragments inserted into
Plasmids.
that's it. Plasmids. Plasmid. Right? And this fragment attached with help of which enzyme? Which enzyme involved here? By attaching, joining of DNA to another gene. Enzyme. Cutting enzyme, restriction endonuclease. Joining
Ligase.
enzyme.
Ligase.
Right. Ligase enzyme. With help of ligase, both are joined. Now next. Now this is this gene called as hello?
Sir.
Plasmid gene and another fragment. Foreign fragment. Both are present, right?
Yes, sir.
Now this is called as
Recombinant, sir.
that's it, rDNA Next step, this rDNA. The rDNA transfer into the
Yes, sir. Recombination. Sir.
yes, this
Bacterial
recombinant
cell,
DNA.
bacteria cell.
Right,
Host
that's it.
cell.
Bacterial cell. That means where these are stored. Where here these rDNA, recombinant DNA stored. Next step, petri dish, agar media, nutritional agar media. Now this bacteria, now this group of bacteria with transferred cell, transferred plasmid, culturing in this. Later, next step,
Screening.
screening. Screening normally which method follow?
Selecting the desired genome, sir.
Yes, selected desired genome by what method?
Colony hybridization.
Right. Same method followed, the same method continued. So which method followed the genomic library? The same method followed here. But what is the different here? Here the main difference of genomic. Can you tell the main difference between genomic DNA libraries and cDNA libraries?
Sir, here non-functional genome we won't take. We'll take only 1% which is present in the DNA.
Okay.
It breaks into fragmentation.
Right. First, let's see the advantages, disadvantages of genomic library and cDNA library. Later, the compare with genomic and cDNA libraries. First, genomic libraries advantages. Let's see the genomic library advantages. Right. What are the advantages? Helps in identification of clone encoded particular gene of interest. It is useful for prokaryotic organism. It is useful for prokaryotic. Right. It is useful for prokaryotic organism because in prokaryotic organism no introns. That's why it is most useful for prokaryotic organism only. And genomic libraries from eukaryotic organisms are very important to study the genome sequence from particular genome because we have to study the entire gene, it including regulatory sequence, introns and exons. Introns also studied, exons also studied. The main disadvantages of genomic libraries from eukaryotic having very large genome content.
It takes long time.
Yes, because if it is prokaryotic, no problem. Because in prokaryotic, there are no introns.
Yes, sir.
That is beneficial advantage. But in genomic library, if you take eukaryotic genome, you collected the entire genome apart from the introns, junk genes also.
Yes,
which
sir.
does not code for protein and also contain non-coding DNA, such as repetitive DNA also present, regulatory regions, which makes them less than ideal. Because actually, if you take non-functional genome, what we do? The genomic library from eukaryotic organism will not work if the screening method requires the expression of gene.
Characterization of genes.
Yes. So these are the disadvantages. Let's see the cDNA advantages and disadvantages. Best advantage is we selected only the functional groups only, functional genomes, not all, right? And we can easily understand the gene expression because you studied the mRNA only. This mRNA later converted into, after, of course, if this formation as a DNA strand, cDNA, reverse transcription process, after formation number of DNAs with help of in normal transcription process, here again the DNA convert into mRNA, right? And mRNA convert into protein. The protein express the enzyme. So that's why the cDNA library is collection of actively expressed gene, but not in genomic library. Because genomic library, junk genes also present, they never expressed the character. So this actively expressed gene in the cells or tissue, right? And second one, already told you, introns not cloned in cDNA library. That means completely reduce the total amount of DNA, which means we can easily handle and easily insert it into the vector, right? The most important application of cDNA library is to study the expression of mRNA, right? Because why the only study of the expression of mRNA? Because what is our template stand in cDNA? Hello? 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 Hello?
Sir.
Are you follow me?
Yes, sir.
The most important application of cDNA library, study the expression of mRNA. What is our template stand in cDNA? RNA is the template. That way easy to express easy to understand mRNA sequence.
Yes, sir.
And disadvantage of cDNA libraries are difficult to create if a source tissue with an abundant mRNA for the gene is not present. That means we know what is luxury genes. There's one small difference here. In genomic library, the entire genome same in every cell. For example, this is an organism. This is an organism. In this organism, you selected the entire gene, entire DNA, this cell. So all cells are identical or not? Hello?
Yes, sir.
Yes,
All
sir.
cells identical genes. What about the cDNA? In
We'll get the
cDNA, for example, let me take two different cells. One is, tell me any two different cells in human beings.
sir, in blood we'll have different cells, sir.
On blood, another cell. Blood
Nervous
cell,
cells,
okay.
sir. Blood
Nerve
cells,
cell,
sir.
okay.
Nerve cells.
Okay, right. Nerve cell, two, enough. Blood cell genome, nerve cell genome content same. Genome same. But this DNA prepare mRNA. Prepare protein. Same way here also DNA, mRNA, protein. Common DNA.
DNA.
What about RNA?
Same, sir.
Protein.
Same.
That means here, this is, do you want hemoglobin? Hemoglobin protein. If you select a nerve cell genome, can you get hemoglobin protein?
No, sir.
That's what here the problem.
Okay, okay. Okay, sir.
Here,
Understood.
this, for example, you selected this mRNA. This mRNA. This mRNA, you prepare from this mRNA, prepare the DNA. After getting from the transcription, translation, get protein. Can you get the hemoglobin character?
No, sir. No, sir.
No, that's what here the different. That mean you have to select it from cell to cell. cDNA, for this you have to select not entire genome, you cannot select. This is different to cell. That is somewhat difficult, right? Introns are absent, hence frequency dependent to the level of gene expression is no need. So this is the major disadvantage of cDNA. Is it clear?
Yes, sir.
Right. Now comparison. In page number clearly given, page number here. Genomic library versus cDNA library. 149. Genomic library versus cDNA library. The genomic library is a large of clones. What about cDNA?
No cloning done.
Of course, clones, but large of clones are fragment genomic DNA represents the entire genome of species. What about cDNA library? Just large collection of cDNA copies, but not all. That means the major difference already told you, only the functional group, functional genomes we selected in the form of cDNA. Okay?
Okay, sir.
A genomic library contains both the coding, non-coding region, right? Hello?
Yes, sir. Yes, sir. Listening, sir.
What about cDNA? Only coding sequence in the form of exons only. Every cell in the in genomic library, every cell in the body has the same DNA. Already just now explained. Just now
Yes,
explained.
sir. Understood.
Same DNA. What
Same
about here?
characters.
What about cDNA? Is it same or different?
Up to formation of protein is same, sir.
That
But
means actually
the basic enzyme formation to which the cell.
my question is, for example, this is the nerve cell.
అంటే నర్వ్ సెల్ కి బ్లడ్ కి ఏదైతే బేసిక్ గా కావాలో అది ఫామ్ కాదు సార్ ఇక్కడ సిడిఎన్ఏ.
నేను అనేది ఏంటంటే ఆ ఎంఆర్ఎన్ఏ కూడా ప్రిపేర్ కాదు.
ఓకే, దానికి సంబంధించిన ఎంఆర్ఎన్ఏ ప్రిపేర్ కాదు.
ఇప్పుడు నువ్వు సిడిఎన్ఏ తీసుకునేది ఎంఆర్ఎన్ఏ కదా.
ఓకే సార్.
ఇప్పుడు ఇదే ఉండదు. ఇప్పుడు ఆ క్యారెక్టర్ సంబంధించిన టెంప్లేట్ లేదు కదా, నువ్వు ఎలా తీసుకుంటావు?
ఓకే,
అదే ఇప్పుడు నువ్వు జీన్ అనుకోండి, జీన్ ఉంటది, దానికి సంబంధం లేదు. ఇప్పుడు ఈ నర్వ్ సెల్ ఉంది, నర్వ్ సెల్లో హిమోగ్లోబిన్ జీన్ ఉంటుందా ఉండదా?
జీన్ ఉంటుంది కానీ హిమోగ్లోబిన్ ఫార్మేషన్ కావాల్సింది ఉండదు.
అంటే డిఎన్ఏ నుంచి వచ్చే ఎంఆర్ఎన్ఏ సింథసైజ్ కాదు. Okay? Clear?
Yes, sir. Yes, sir. Clear, sir.
In genomic library construction, restriction endonuclease, restriction enzyme, DNA ligase enzyme used. In cDNA, both used. Same restriction enzyme used in cDNA for cutting, for joining DNA ligase used. Apart from that, two extra enzymes also utilized. One is reverse transcriptase, another DNA polymerase. What is function reverse transcriptase? Reverse transcription from the RNA to synthesized DNA. DNA polymerase enzyme, one single stranded DNA becomes double stranded. That's why extra two enzymes utilized in this. And again repeated, the genomic library will be very unlikely to contain the entire coding region, but here cDNA no, only introns are removed, exons present. Genomic DNA libraries provide much more detailed information about the organism because we selected the entire genome. cDNA library only lack of non-coding and regulatory elements found in genomic DNA. However, the information of cDNA library is powerful, useful since the gene product are easily identified. We cannot take entirely, but we can take only active portion of gene, right? So these are the differences between genomic library, cDNA library. Very, very important in theory and comparative. cDNA genomic library. Clear? Hello?
Sir.
Yes, sir.
Yes, sir.
Means we have to write the differences, disadvantages, and comparison.
First,
Please.
actually, just that is based on question.
Okay.
For
Okay,
example,
sir.
if they give the long question, write about genomic libraries. First, you have to start with definition. Whether they ask or not, you should start with definition. A brief introduction about it, if any brief introduction. Next, either two methods. In genomic library, two methods. One is construction, another one is screening. Later, advantages, disadvantages. Enough. That's enough. If they ask the question as a write about genomic library and compare with cDNA libraries. If that's so, you have to write the comparison also. Okay. In the same way, cDNA also. In cDNA, if you ask the cDNA, just write about construction, screening. If they ask comparison, just compare with genomic library. Is it clear? Hello?
Yes, sir.
Right. Next one, polymerase chain reaction. This is also very, very important question in theory and the competitive level. What is polymer chain reaction? Polymerase chain reaction is a laboratory technique used to make multiple copies of segment of DNA. That means we want to get more copies. If you take the one DNA, it get into number of copies. The polymerase chain reaction developed in 1983 by American biochemist Carey Mullis. He got Nobel Prize in 1993. Using PCR, it is possible to generate thousands to millions of copies of a particular section of DNA from very small amount of DNA.
Sir, cloning has started from that year only, no, sir? Cloning.
Cloning,
Sir.
cloning, first clones.
First clone is done on sheep. Sir.
I have no idea about the cloning,
Okay,
but as far I know, before 1983, I think 1960,
1960.
cloning, but I have no idea about it.
Okay, sir. Okay.
But I am not sure. I will tell you next class.
I'm also not sure, but I have a little bit idea, sir. So I'm asking.
Okay.
Okay, sir. Okay.
Right.
Dolly, mommy.
Principles, and that means the main requirement to the polymerase. To get number of copy from small amount of DNA, here some are the requirement, or some of the requirement. One is DNA template, DNA polymerase, oligonucleotide polymers, deoxyribonucleotide triphosphate, buffer system. They are all required. First, let's see DNA template. If you want to get number of copies, there is the first one double-stranded DNA present, right? Without DNA, how can make a number of copies? Is it possible? Hello?
No, sir.
No, sir.
That's why we should need one double-stranded DNA, right? After that, double-stranded DNA from which enzyme require from the synthesis of new DNA strand?
Reverse transcriptase.
No. We want a DNA only. I am not asking about reverse transcriptase. Okay, in cDNA library, you opted reverse transcriptase. After getting DNA, to make another DNA, which enzyme you using?
Polymerase, sir.
Polymerase, that's it. That is the second one, DNA polymerase.
Okay.
But especially in DNA polymerases, another TAC polymerase is also utilized. What is the importance of TAC? Already explained about it.
Resistant to heat.
Right, that's it. Resistant, because in PCR chain reaction, we can use the temperature, high temperature. To resist high temperature, we can use TAC polymerase. Next, oligonucleotide primers. This is the boost into DNA polymerase enzyme. Without primers, remember, without primers, DNA polymerase never do this activity. When you add the primers in the form of start stretches single-stranded DNA, if you add to DNA polymerase, then only they start the activity. The start stretches single-stranded DNA complement to the 3 end of the sense antisense strand. 3 end of the both sense strand and antisense strand. Next one. The two separate in oligonucleotides, two separated strand of DNA are complementary and run in opposite direction. From one end, one end, the 5 end, and to other end, 3 end. As a result, there are two primers, a forward primer and reverse primer. Here the primers, two types. One is primer, one direction, another one is reverse direction. That's why here, where is it? Because here, just wait. Five end, 3 end. Opposite should be 3 end, right?
5
end. Okay. For example, this is called, in this both, one is sense, another one is antisense, right? One stand sense stand, another stand is antisense stand. If you two primers, one primer, add it. Always the primer add to 3 end only, remember it. Okay. Now the next time this is 5 end. Opposite 5 end all. Is it okay? This is 3 end. Newly synthesized always. Newly synthesized strand DNA direction always 5 end to 3 end or 3 end to 5 end. Synthesizing DNA direction.
5 prime to 3 prime.
That's it. 5 prime to 3 prime. So here this is 3 end, right? Opposite should be 5 end. 5 end, right? Opposite should be 3 end. Here 3 end. So here opposite. So this is reverse direction. Back. Here this is 3 end and primer attached here. So here 5 end. Forward. Both directions. Right? Is it okay? Primers clear?
Yes, sir. Yes, sir.
Right. Complement to the 3 ends. 3 end of sense and antisense.
Sense, antisense, strands.
Deoxyribonucleotide triphosphate. Provide energy for polymerization. What is polymerization? Building blocks for DNA synthesis. Means here, for example, this is one strand. This is preparing strand. Already here given. 3 end to 5 end. 5 end to 3 end. Already 3 end here. A.
It's all monomers join together, sir,
Yes,
to form polymer.
that's it. G, T. Now with the joining T, A, G. This activity done by deoxyribonucleotide triphosphate. Deoxynucleotide triphosphate. Is clear?
Yes, sir.
A buffer system. Magnesium, potassium in buffer system. Magnesium, potassium both are provide optimum condition for DNA denature and renature. What is denature? And also involved in polymerase activity. Sorry, polymerase activity. Polymerase activity done by deoxyribonucleotide triphosphate. And also buffer system. Denature, separate, renature, again winding. Winding. Actually, DNA nature always winding with double helix, right? Winding with double helix. So denature means rewind, separate. Again unwind. And rewind, that is renature. Clear?
Yes, sir.
So these are the principles requirement of the PCR cycle. Let's see the procedure. Procedure PCR. చాలా సార్లు అడిగారండి క్వశ్చన్స్ రిపీటెడ్. So there are three steps in PCR procedure: denature, annealing, elongation. In denature, mixture means here already DNA content mixture. Heated here first. (94°C). Heated up to 94°C. About 5 to 2 minutes. Due to heat, the break is hydrogen bonds. Where are the hydrogen bonds? Where are the hydrogen bonds? The hydrogen bond between the two strands of DNA. As a result, convert into single-stranded DNA. This process called denature. That means denature, double-stranded DNA becomes single-stranded DNA. That's it. It takes 15 to 30 seconds. The single stranded now act as a template. Each single stranded act as a template for production of new strands of DNA. New strands of DNA, right? High temperature causes hydrogen bonds between the bases. Two strands break into separate. Next, annealing. In annealing stage, the reaction temperature is lowered first here 94, then down into 54 to 60 degrees centigrade. For around 20 to 40 seconds, this reaction. Now in this annealing, primers added. What is the function of primers? What is the function of primers? Already
New
they explained
DNA,
in
sir. New DNA. Yes.
synthesizing DNA with help of how new DNA synthesized? Which
With
enzyme
the
involved?
polymerase, sir.
Which polymerase? There are number of types: RNA polymerase, DNA polymerase.
DNA polymerase.
DNA polymerase, right? The primer uses single stranded of DNA. The primers are digested the complementary sequences, short sequence section of DNA of each end of the sections. That means each end means three end, mainly three end. The primers, where it is, actually the primers serve as a starting point of DNA synthesis. Now, not started yet. After added the primers, after adding, where is this polymerase enzyme? Primers, right? Polymerase, DNA polymerase attached. So start making new complementary strand. As you said, the requirement of synthesis DNA polymerase, but here the utilizing TAG DNA because if normal DNA denatured in this temperature, that's why use the TAG DNA polymerase enzyme. Right? Is it clear? Hello?
Sir.
Now elongation. That means here, in what happened after separate into two steps, one DNA strand, two DNA strand. For example, this is 5 end to 3 end. Here it is actually the opposite. Opposite means 3 end to 5 end. Here primers attached. Here primers attached. Here DNA polymerase attached. Here DNA polymerase attached. Now ready to start. What are the requirements for polymerization? In elongation means elongate the DNA in the 5 end to the 3 end. This is very, very important. 5 end to the 3 end direction. Here the primer, right? Hello? Are you follow the marker? Cursor?
Yes, sir.
Yes, sir.
3 end. This is the 3 end. Now newly synthesized, I already told you many times, 5 end to 3 end. Here this is 5 end. 5 end to 3 end. Right? This is the 5 end to 3 end direction. Temperature raised up to 70 to 80 degree. Temperature raised up to 70 to 80 degree. Here 94 degrees centigrade. Here 50 to 60 degrees centigrade. Here 80 degrees centigrade. These 3 steps are repeated 20 to 40 times in order to obtain number of sequences of DNA. Is it clear? Procedure clear? Elongation or extension. These 3 steps: denature, annealing, elongation. Hello?
Yes, sir.
Any missing in requirements? What is this? Actually, this elongation done by polymerization of DNA, right? The polymerization activity done by which enzyme? Which requirement? Not enzyme here. The which enzyme? Deoxynucleotide triphosphate. Deoxynucleotide triphosphate added the nitrogen bases to the synthesizing DNA. Right? Clear?
Yes, sir.
Next one. If you clearly understand about PCR, next steps you can easily understand. That is real-time PCR, RT-PCR, so on. Is it clear first?
Yes, sir.
Right. Once again, let's see in diagram. Right. Oh, shit. Are you able to see the screen? Hello?
Sir.
Yes, sir. We are able to see, sir.
Denature, 95 degrees centigrade, separated. Right? Add primers. So which end primers are added?
5 prime to
3 prime. This is synthesizing stand. 5 and 2, 3 and synthesizing stand. Here added 3 prime. Okay? Both sides 3 prime. An extension before extension, this is this one. DNA polymerase added. TAC polymerase. Apart from TAC polymerase, here deoxynucleotide triphosphate added to this strand. Now extended into double strand. Later, this is again converted into two strands. Multiple, like this, multiple copies are formed. Number of copies are formed 2 to 40 cycle. 2 to 40 cycle, we get number of content of DNA. Clear? Okay. Now, real-time PCR, RT-PCR. Next our book. Next, what is the next one? One which annealing completed and extension. Okay, these are completed stages. Application of PCR. Application. Before going to know about application, diagnostics, and other forensic studies, evaluation studies, just know the mechanism of PCR. Real-time PCR and RT-PCR. Don't get confused with these two words. One RT-PCR is real-time. Another RT-PCR reverse transcriptase. Reverse transcriptase PCR was real-time. In real-time, same we know the PCR steps in normal PCR steps. Denature, annealing, extension. These all steps done in the separate instrument that is called thermocycler. For example, this is thermocycler. Thermocycler. Just wait. I don't want to waste your time and our time. This is thermocycler. Thermocycler instrument. In thermocycler instrument, this RPCR reaction takes place. And this thermocycler instrument connected with another separate device. In this device, you can measure the total content of DNA. Total content of DNA. So this is called quantity. You can select the quantity of DNA. So this real time called as qPCR. Q for quantity. For example, thermocycler, you repeated into number of time PCR cycle, but you don't know how much content of DNAs are formed. Do you know without this device, the spectro machine or spectrophotometer machine, UV spectrophotometer, which detects, deflects the shows the number of content. After 20 cycle, after 30 cycle, after 40 cycle, how much content of DNA are formed reveals it. That's why this is called real time. Real time or qPCR. Is it clear first? Hello?
Yes, sir.
Clear first one? In normal PCR, only with help of thermocycler device, you sample of DNA, multiple number, it makes into multiple number, but you don't know how much content of DNA you get. But when you connected this thermocycler device to UV spectrophotometer device, automatically this screen show you how much content of DNA, 10 grams, 40 grams, 50 grams. So this content of DNA reveals, explain about it. That's why it's called as a real-time or qPCR. Next one, reverse transcriptase. What you take in normal PCR? Hello? What did you take in normal PCR? DNA or RNA?
DNA.
DNA. But in reverse transcriptase, RNA, reverse transcriptase enzyme now converted into DNA. Reverse transcriptase converted into DNA. Now they make into number of copies. Same process. Process same. Copying number of copies into DNA copies. By denature, by annealing, by extension. Right? This is RT-PCR. You must have listened about RT-PCR test for COVID, right? Hello?
Yes, sir.
Yes, sir.
In RT-PCR test COVID, what we get, for example, this is COVID infected person. COVID infected person. Through snap test. Do you know snap test? This is the person. Right. Person. The diagram, the real-time PCR, the picture this time ledu, and the diagram చూసుకోండి. Snap తీశారు. ఓకేనా? What are present in snap? Nose ని snap తీస్తారు కదండి. Take the snap with nose. Okay?
Okay, sir.
Right. Collected with nose. And these are taken into the tubes. Actually, this COVID virus genetic material RNA. It may be RNA for some virus detection, some bacteria detection, any one, maybe RNA or DNA. If it is DNA present, directly take the DNA mixture, eat it, denature, annealing extension. Clear. If it is RNA, through RT-PCR. RT-PCR means this RNA through reverse transcriptase enzyme convert into DNA, right?
DNA.
Now this DNA, actually this formed single stranded. This single stranded with help of polymerase enzyme convert into double stranded DNA, right? Now this through the polymerase chain reaction convert into number of copies. Number of copies. Okay. So clear? Are you clear so far?
Yes, sir.
Right. But how to get whether it is positive or negative? Positive, negative. That is done by just assume. How can decide positive or negative? This
RNA
content
enzyme.
and this content, actually this PCR cycle content done in which instrument? Instrument name already told you.
Thermocycle.
Thermocycler, right? Thermocycler, okay. The thermocycler device connected to?
Spectrophotometer.
Right. Spectrophotometer device, UV spectrophotometer device, right? What is the function of this? Reflect the, it shows the content of DNA. Content of DNA. Total content of DNA. Right? Clear? Just wait. CT value. Through CT value, we can identify positive, negative. Simple. Just one thing. For example, this is patient A. This is patient B. A person, not a patient, A person, B person. You collected snap from the A person and the same B person. Through the PCR, decided the content. And also PCR cycle also. PCR cycle also, remember, PCR cycle also. But here the A person to get, for example, our convenience, to get 20 grams DNA, 20 gram, the viral DNA, to get 20 gram viral DNA, you take 20 cycle, for example, 20 cycle. But B person take 40 cycle. Even though he take the 40 cycle, he get only just 10%, or he cannot found any gene sequences, any gene content. Who is positive? Who is negative? A or B? Hello? Hello? Is it audible to you?
Yes, sir. Yes, sir.
A.
Right. That is called CT value. That is called CT value. The optimum CT value cycles 30. 30.
Oh,
If you get desirable content of gene below 30, below 30, below 30 means maybe 10th cycle or 20 cycle, 25 cycle. That means actually in PCR, one DNA strand prepare 2 DNA one cycle, 2 to 4, 2, 4 to 8, 2, another cycle. Like that, number of cycles are there. Like that, if reach the 20 cycle, the more content of DNA already with help of this device, we estimate the total content of DNA, right? If the total content of DNA, if we estimate, we consider as a positive. If it is the same content or just below the content of DNA formed 40 cycles, take 40 cycle. In this device, we can see the number of cycles and total quantity, content of the DNA also. If the cycles exceeded 30, exceed 30, considered as a negative. Of course, the content formed, different genome content formed. Here, the which genome here are you collected? Did you collect here? Snap from the nose. That means the virus. What is present our nose? If we infected with coronavirus present in nose, right?
yes.
That viruses, for example, he infected heavily. That means he already 10 virus present. Be infected in general, just only 2 virus only there, which is high content. Which is high content? A or B?
A.
A high content. Which is give more content, less cycle, less CT cycle. Less CT cycle, production of high content, positive or negative?
Positive, sir.
Positive. More CT cycle, less content. More CT cycle, more means more than 30 CT value, less content. Negative or positive? Hello?
Negative, sir.
Right. That's it. Are you clear? So this is the diagnosis method, not only viral, that means not only coronavirus, but recently the rapid గా ఇంక్రీజ్ అంటే ఇప్పుడు మనకు ఎక్కువ న్యూస్ లో వస్తుంది ఇదే కాబట్టి దీని గురించి ఎక్స్ప్లెయిన్ చేయాల్సి వచ్చింది. మిగతా ఏ డిసీజ్ అయినా సేమ్ ఇంతేనండి. ఇక్కడ డిసీజ్ ఇచ్చినా నో, ఇంకేం డిసీజ్ ఇవ్వలేదు. ఏదైనా సేమ్ ప్రాసెస్ ఇంతే. ఫర్ ఎగ్జాంపుల్ టైఫాయిడ్ ఉందనుకోండి, ఫర్ ఎగ్జాంపుల్ టైఫాయిడ్. So here the typhoid genes, they are all typhoid genes. Number of content of typhoid gene decided as a typhoid. HIV virus, number of content of HIV virus genome, that is considered as a positive. Clear? Hello? Hello?
Yes, sir.
Yes, sir. Clear.
Any doubt today lesson?
Wow.
All right, can I leave?
Yes, sir.